Now, Paul Feeney's unique contribution to this literature was to draw the analogy between this reciprocal adaptive evolutionary process and the co-evolutionary arms race that characterized the Cold War at the time it was proposed, that reciprocal evolutionary change between attacker and attacked leads to increasingly deadlier weapons. So I went to Cornell for graduate school to work with Paul Feeney, and there I met two other Tyler Prize winners who were very influential on my career. Tom Eisner, whom you just heard, who just died a couple of weeks ago, a tremendous loss for the whole, well, the world of humanity, not just the world of science, and Gerald Meinwald, who are together really elucidated the ways in which insects use chemicals to defend themselves against their enemies. So ecological chemical warfare. It was a major focus of many labs at Cornell. So it was inevitable that, again, I would have to deal with things that can kill you. So I moved on not being afraid of insects, and I would not be afraid of chemicals. So I knew in the Feeney lab I had to work with chemicals, and I had to pick a chemical. And Paul, in fact, handed me a book called Biology and Chemistry of the Umbelliferæ, an edited volume by V.H. Haywood, and he told me, if you can find a chemical in this book, I can support your research. So that sent me reading the book, and fortunately there was a language requirement at Cornell, so I could read this French article about a group of incredibly nasty chemicals called phenocoumarins. They were fascinating, largely because were unbelievably broadly biocidal, toxic to just about everything. Their mechanism of toxicity had been worked out. In the presence of ultraviolet light, these chemicals are capable of absorbing light energy to form what's called an excited state molecule that then becomes highly reactive. And the way it reacts is to bind to pyrimidine bases in DNA. If sufficient UV light is present, they can actually cross-link strands of DNA. Now, the problem with cross-linking strands is that it interferes with transcription, the process by which DNA ultimately ends up replicating itself. So phenocoumarins then interfere with the way cells grow, reproduce, divide, and reproduce, and effectively are toxic to anything with DNA, which is life on this planet as we know it. At the time, they had been shown to be toxic to germinating plants, to bacteria, to fungi, and they were well known to be toxic to people. Even casual contact upon the skin caused what's known as phytophotodermatitis, sometimes photophytodermatitis in the literature. And they're so potent, in fact, that their ability to photosensitize and then induce a hyperpigmentation response was developed medicinally in the 1970s for use as a therapeutic agent. The procedure called PUVA, which is still in use today for psoralen plus UVA, silent P, is used for treating various skin disorders, including vitiligo and psoriasis. It's very potent chemicals, but no one had actually determined what their impacts might be on insects. And that's one of the first things I did, was to see, in fact, that these are highly toxic to insects, particularly insects that feed on a broad range of plants. So the southern armyworm, for example, has many dozen different host plants, but scrupulously avoided plants containing phenocoumarins with good reason, because at the parts per billion level, they're highly toxic. And the toxicity appears to be associated with this extra ring, this furan ring, because the precursor chemicals, which are widespread in plants, was not toxic. So this, to me, seemed like a really interesting example of escalation in the arms race, where just simply adding a third ring to this chemical allowed the cross-linkage, which causes a lot more toxicity than the precursor chemicals are capable of. But that's not coevolution. That's just a toxic chemical. What I needed then for the next spin of the cycle was to find insects that could cope with these chemicals, because that's what the Ehrlich and Raven scenario would predict. So here's where fear enters in again. I'm afraid to drive. Another thing my sister will attest to. I don't like driving. There are even parts of Champaign I won't drive in, even after living there for 35 years. And as a graduate student, I didn't want to drive. So I had to find insects that feed on plants with phrenocomerins within walking distance of my office. And there's my office. Well, you see the bee right there. So I started walking from the insectary and ultimately came to a large field, it's not there anymore, of these plants, wild parsnips. It's now just a grass. This is the equine metabolism unit. There used to be ponies in here. I had no idea that was the equine metabolism unit. And I used to feel sorry for them because they were in concrete pens, and I used to give them clover and other things. I think I must have screwed up a few dissertations unknowingly because I felt sorry for the ponies. So I found a population of parsnips. So I had it made if I could find insects eating these parsnips. So this is Pastinaca sativa. It was not it's the escaped version of the not too popular vegetable plant. There's a long history. Actually, parsnips were planted at as early as 1609 at the lost colony of Roanoke. Colony was lost. I don't think parsnips had anything to do with it, but nonetheless, cultivated plants have escaped all around the world where parsnips are grown to become noxious weeds. I mean, the vegetables are kind of inoffensive, but the weeds are truly noxious. And that noxiousness is associated with the fact that all above ground parts produce 6 different phrenocomerins. So what they do is cause this photophytodermatitis. There's my hand there. And there are warning signs many places that, again, casual contact with the above-ground parts of the plant produce this painful, unsightly, oozing, blistery rash that takes a long time to heal and ultimately leads to hyperpigmentation of the skin. Now, given the warfare metaphor, it's really interesting that the photodermatitic properties of parsnips were discovered during World War II. This was a study published by John Belisario with the Australian General Hospital in New Guinea, who published a paper reporting his examination of 9 personnel admitted to the hospital suffering from skin manifestations on hands, arms, and trunks, suspected of having been caused by mustard gas dropped by enemy planes. The actual cause was quickly determined, however, to have been peeling parsnips. So, again, an international disaster was averted by an astute insight on the part of Dr. Belisario. As a result of his studies of the blister-inducing properties of parsnips, Australian troops in New Guinea were ordered to peel parsnips indoors, or if peeling outdoors, to do so in the late afternoon or evening by personnel fully clothed, the sleeves rolled down, and preferably wearing rubber glove on the hand which holds the parsnip is dipped in water. So parsnips go to war here. So there were, in fact, insects on parsnips. Most of the insect, not many, and parsnips actually has a pretty depauperate insect fauna compared to most other weeds, at least in part because of these phrenocomerins. Most of the insects were associated with the leaves and the stems, but I was astonished to see repeatedly, and in almost every plant in the population there, an insect infesting the flowers and fruits, which are up to 10-fold higher in these DNA cross-linking toxins. So who is this insect? Is Deprecaria passinacella, the parsnip webworm. It is a caterpillar that feeds, doesn't just feed on phrenocomerin-containing plants, it feeds exclusively on phrenocomerin-containing plants. Only 2 genera in the carrot family. One is parsnip pastinaca, and the other is a very closely related genus, which actually some systematists think maybe belongs to the same genus. Every day of its life, every mouthful of its food contains phrenocomerins. And